In this video, I'm going to introduce you to glutaminergic synapses. These will be synapses where the neurotransmitter glutamate is released. We need to look at the events that occur at this synapse in some detail. So here's my blow-up of a glutaminergic synapse. I've drawn the terminal on the left in green, and then I have the target cell on the right. And here I've drawn it as a dendritic spine because in the central nervous system, glutaminergic synapses often occur on spines. That's not the only place we can find them. We can also find them on spineless dendrites. We can find them on axon terminals. There are even some glutaminergic inputs onto cell bodies. The other thing I'll add to this picture will be an astrocyte. That's a type of glial cells, and in the central nervous system, synapses are intimately associated with glial cells that help to regulate the concentrations and recycle some of the players. Arguably the most obvious specialization of the axon terminal will be these synaptic vesicles, which are filled with glutamate. And again, that's the major neurotransmitter at excitatory synapses in the vertebrate brain. The postsynaptic cell will also have specializations. There'll have to be receptors for glutamate, and there'll be some reuptake transporters in order to recycle the neurotransmitter, and those can be found both in the axon terminal as well as in the glial cells. Well, now what I want to do is zoom in on events in the axon terminal, and we're going to look step by step at what it takes to load vesicles with glutamate, release them, and then recycle them. Well, the neurotransmitter at glutaminergic synapses is glutamate. It's a small molecule. It's an amino acid. The molecule can be synthesized in the cell. In the mitochondria, we can also take it up from outside the cell. Either way, once the glutamate's in the axon terminal, we need to pack it into the vesicles. That's going to involve two membrane proteins. There's a vesicular glutamate transporter and then also a vacuolar ATPase pump. So the vesicular glutamate transporter, vGlut1 and vGlut2, there are two different versions of this, are antiports. They move glutamate into the vesicle interior against the concentration gradient, and they pay for that by allowing a proton to move down its electrochemical gradient. In order to create the electrochemical gradient for protons, the cell is constantly running this vacuolar ATPase pump, breaking down ATP and then moving proton into the vesicle so that the interior of the vesicle is highly acidic. Release of the vesicle is going to involve docking with V and T snares. This will be triggered by the influx of calcium, and when that happens, then the contents can be released. The transmitter can diffuse across the membrane, bind to its receptors, and then the vesicle itself can be recycled through membrane endocytosis, where it can then be reloaded with neurotransmitters, so this process can continue. Well, let's turn to the postsynaptic side and see what happens there. There are a number of different types of receptors for glutamate, and I'll just show some of them here. Some of them are ionotropic. That means that the receptor protein itself is an ion channel, and we see a few examples here. Some of them are simple ligand-gated channels, which will open up and allow the influx of usually sodium into the cell. That would be the AMPA or the kainate class. In glutaminergic synapses, there can also be a second type of ligand-gated channel. This is a little bit more complicated because it has a binding site where it can bind glutamate and open up, but there's an additional magnesium block which needs to be removed in order for ions to move, and I'll show you more about that in a little bit. In addition to these ion channels that act as receptors, glutamate also has what are called metabotropic receptors. Metabotropic receptors are not ion channels. They're members of what are called the GPCR, G protein coupled receptor family, and what they'll do is when they bind glutamate, they'll initiate an intracellular cascade that can lead to events in the cell. There are several different varieties, and we lump them into these three broad classes: metabotropic glutamate receptors 1 and 5, 2 and 3, and then 4, 6, 7, 8, and they get linked by what the intracellular targets are that they activate or inhibit. Well, I want to talk about these in just a little bit more detail. So let's begin by looking at the ligand-gated ion channels, these ionotropic receptors. These are pore-forming channels, and they're going to allow the passage primarily of sodium, although some other positive ions can come in. They open when glutamate binds, and the influx of positivity then will make the cell more positive. It will depolarize the cell. So let's see how this happens. In the absence of glutamate, the receptor will be closed. If we add glutamate, the glutamate can bind to the binding site. That will open up the binding gate, and now sodium flows into the cell. It's a very rapid response. NMDA receptors are a little bit more complicated, so let me walk you through those. They're also pore-forming membrane proteins. They allow the passage not only of sodium but also of calcium, and this is different from the other glutamate receptors that we'll see. Activation of these channels by glutamate depends on two factors. There's a ligand gate that will open when glutamate binds to the channel. There it is right there. And in addition, there's a magnesium block that here I've indicated by a red star. This needs to be removed in order for the ion channel to allow the influx of sodium and calcium. In the absence of any glutamate, then the channel will be closed. There's nothing bound to the ligand. Sodium can't enter. Even if we go and we add some glutamate, the glutamate can come and it can bind, and that will open up the ligand gate, but there's still no way for the ions to move through because of the magnesium block. However, if the cell is depolarized, it gets more positive, then what can happen is that will remove the magnesium block. And now when glutamate comes and binds to its binding site and the ligand gate opens up, sodium and calcium can flow into the cell. Activation of NMDA receptors can allow calcium to enter the cell, where it can modulate the activity of a number of different proteins. It can act as a coincidence detector because this channel will remain closed unless the magnesium block has been removed. Let me show you what I mean by that by returning to the synapse. Well, here I've got a postsynaptic cell that's getting input from two glutaminergic synapses. If there's an action potential in presynaptic input 1 and it arrives, well, it will release glutamate, and then those ionotropic receptors can open up and sodium can enter the cell. But there won't be any influx through the NMDA receptors because the cell hasn't removed the magnesium block. Well, now suppose what happens is we first activate presynaptic input B. Sodium will enter through that cell, depolarizing it, and in addition, that influx of sodium can cause the magnesium block to leave the channel. Well, now if there's an action potential in input A and glutamate gets released, not only will there be sodium influx through the AMPA and kainate class of ion channels, but as long as the magnesium block hasn't had a chance to go back in and plug up the pore, we'll also have an influx of calcium through the NMDA receptors. This increase in calcium inside the cell when both synapse A and synapse B are activated together can be really important in memory and learning. It will allow a way for that gray neuron to know that those two inputs are associated, and it can cement that using calcium as the signal to implement those changes. I want to say a word about the other type of glutamate receptors, the metabotropic receptors. Now remember, these are integral membrane proteins, but they're not pore formers, they're not ion channels. They'll change configuration when the ligand binds. They're associated with what are called heterotrimeric G proteins. So this is a set of 3 small proteins which are associated with it. And in this state here where it's off, the heterotrimeric G protein will be associated with a molecule of GDP. What happens when we add glutamate is that the glutamate binds to the receptor. That induces a conformational change, which in turn leads the G protein to drop the GDP and instead pick up a GTP molecule. And once this happens, the subunits of the G protein lose the affinity for the receptor. They'll diffuse away. And in the case of the glutaminergic receptors, the alpha subunit can go and interact with a number of different effector proteins in the cell. They do different things depending on the particular type of glutamate receptor that was activated, but in general what happens is there's a cascade of events in the cell, and we might, for example, phosphorylate an enzyme, activating it, and that activated enzyme then could go and have other activity in the cell. Effects that are mediated by metabotropic receptors are slower and longer lasting relative to the ionotropic receptors. So what we've seen is that at glutaminergic synapses, the glutamate release will depolarize the cell, it might activate NMDA receptors and lead to an increase in calcium, and it might also activate signaling cascades that can have long-lasting effects. Well, there must be a way to reset the synapse so it will be ready for the next round of information. This will be accomplished by removing excess glutamate from the synapse using reuptake transporters. The reuptake transporters are called the excitatory amino acid transporter, or the EAAT class of transporters, and there are several different types. We find different versions in the axon terminal and in the glial cell. They operate with a somewhat ridiculous stoichiometry. So for example, some of them will move 3 sodium ions, 1 proton ion, and countertransport a potassium ion every time they want to move 1 glutamate out of the cleft and back into a glial cell. It's not by coincidence that I drew the reuptake of glutamate into the glial cell. At synapses in the vertebrate nervous system, 90% of the glutamate reuptake is actually done by the glial cell. Now, it would be nice if the body had a way to return that glutamate to the neuron so it could be repackaged, and there is. What happens in the glial cells is that glutamate is converted into a compound called glutamine, and then glutamine can be released from the glial cell, taken up by the signaling cell, turned back into glutamate, and then repackaged at the axon terminal for reuse. Well, I hope this step-by-step walk through the events that occur at a glutaminergic neuron will make you appreciate the complex processes that are necessary in order for you to think and learn.